This video is about gene expression. So first, what is a gene? A gene is a small piece of genetic material written in a code and called DNA. Every person has 2 copies of each gene, one from each parent. So our genes are what makes each person unique. It contains the instructions for making the molecules that an organism needs to survive. The genes must be transcribed from DNA and then translated in the ribosomes in order to be turned into gene products like protein that the organism can use. Did you know in humans a gene can be between a few hundred DNA bases to more than 2 million bases long? The Human Genome Project has estimated that we have between 20,000 and 25,000 genes. Moving on to differentiation. Embryonic stem cells are pluripotent and thus have the ability to give rise to any type of cell in the body. When an embryo is developing, the stem cells become specialized for one function or a group of functions. The structure and function is determined by the proteins which are synthesized. The proteins are synthesized based on the cell's DNA, however, all the cells have all the DNA present. Therefore, the cells must only be expressing certain genes, the genes which are switched on for that cell. This is the Lac-Operon model, which explains how genes are controlled in the prokaryote E. coli. Beta-galactosidase is the enzyme produced to break down lactose into glucose and galactose. Now to explain the model. When lactose is not present, a lactose repressor molecule binds to the DNA, and as this diagram shows, it binds to the operator, meaning that the RNA polymerase cannot bind to the DNA promoter region. Since RNA polymerase must bind for transcription to start, the beta-galactosidase gene cannot be transcribed. If lactose is present, it binds to the repressor molecule so that it is prevented from binding to the DNA, and the beta-galactosidase gene is expressed because RNA polymerase can bind, so transcription of the gene starts. Once the mRNA strand has been made, it is translated to produce the enzyme. The process is similar in eukaryotes. RNA polymerase must bind to a section of the DNA adjacent to the gene to be transcribed, which is known as the promoter region. The gene remains inactive unless the enzyme attaches to the promoter region successfully. Only when the enzyme has attached to the DNA will transcription proceed. Also, the attachment of a regulator protein is usually required to start transcription too. Transcription can be prevented in a couple of ways using protein repressor molecules. They can attach to the DNA of the promoter region, blocking the attachment site, and in eukaryotes they can also attach to the regulator proteins themselves. In the first case, RNA polymerase cannot bind to the attachment site, and in the second case, the regulator protein cannot bind, and so in both cases the gene is switched off and is not transcribed within the cell. I will now briefly explain the process of transcription. In transcription, the DNA uncoils and separates to expose the bases guanine and cytosine, adenine and thymine. RNA polymerase adds complementary RNA nucleotides to the template or sense strand. The nucleotides form a strand which is identical to the coding or antisense strand, except the presence of ribose sugar and uracil is transcribed instead of thymine. Earlier on in this video, we mentioned how genes can be switched off using protein repressor molecules. Gene expression can also be controlled using epigenetic markers. In DNA methylation, methyl groups are attached to the DNA of a gene, usually to cytosine, which prevents the transcription of this gene. In histone modification, epigenetic markers such as methyl or acetyl groups are bound on histone tails, which alters how tightly the DNA winds around histone proteins. When the DNA is wound tightly, genes are inactive.